Hey guys, welcome back to One Stop Biology. So guys, in today's video, we are going to continue with the cell cycle and cell division chapter, which is chapter 10 of class 11th NCERT. And today we are going to study mitosis. So we are going to study the M phase. And if you remember in the last video, we basically discussed the entire phases of cell cycle, right? And then basically we discussed all about the interphase. So after that, we are left with the mitosis phase, right? So let's see, what the mitosis phase is, right? So guys, mitosis phase is considered as the most dramatic period, right? And why? So it is basically the most interesting period of the cell cycle, but because it is basically nothing but virtually reorganization of all the components of the cell. So all the components of the cell are basically getting reorganized, right? Now we remember that the chromosome number in parent and progeny cells will remain the same in case of mitosis. What I said is chromosomes in parent and progeny cell is same. And that is why mitosis is considered as equational division. It is basically equal division, right? Because after dividing of the cell as well, the chromosome number remains equal in both, right? So now, if we have to see how mitosis is divided into different stages, mitosis is divided into four different stages, and these stages are prophase, metaphase, anaphase, and telophase, right? So there are four phases: prophase, metaphase, anaphase, and telophase. So let's start with the first phase, which is the prophase. Now, again, this is the first phase, and this is followed by the S and G2 phase. So, if you remember interphase, now interphase had three stages, right? It had G1 phase, which is gap 1, then synthesis phase, where the DNA replication and synthesis takes place. And then we have G2 phase, gap 2. So after the end of that interphase, after the end of G2 phase, we have M phase starting, and M phase starters basically with the prophase. So prophase exactly comes after the G2 phase of interphase, right? So now in G2 phase, we know that the new DNA molecules which are formed by replication, what happens is they are distinct, they are not intertwined, right? So basically, what I mean to say is that once the cell reaches the prophase, the DNA molecules are not intertwined, right? And what happens is that after the cell enters the prophase, the condensation of the intertwining of chromosome material starts, right? So basically, first thing in prophase is that the chromosome material condenses. First is chromosome material condenses, and it forms compact mitotic chromosomes. It forms condensed chromosomes with a centriole, right? So basically, to form compact mitotic chromosomes. So, so if we talk about the structure of this chromosome, what happens is basically they will have two chromatids, two sister chromatids, and they'll be joined together by a centromere. So you'll have two sister chromatids joined by a centromere. So first thing that happens in prophase is that the DNA, newly formed DNA molecules, they condense and they form the compact mitotic chromosome, right? Now, basically, what happens after that, right? So after that, now what happens is that the centriole, which has undergone duplication during S phase, that begins to move towards the opposite poles of the cell, right? So basically, we have what? Second thing is that we have the mitotic spindle forming. So the start of mitotic spindle formation, the initiation of mitotic spindle starts, right? And that happens in the microtubules, right? Now we know that we know what exactly is microtubules, right? So microtubules is a protein component, a proteinaceous component of cell cytoplasm, which helps in this process, right? So what happens is that initiation of assembly of mitotic spindle. Right, so these two things happens in prophase. Now we do not have, organelles like Golgi complex, endoplasmic reticulum, nucleolus, and nuclear envelope present during prophase. Remember that, guys, right? So if you see here the early and late prophase, what is happening is here if you see that the chromosome is not condensed. So it is basically the intertwining has started here in the early prophase stage. You can see that the DNA molecule, the newly formed DNA molecules are intertwining, and by the time we reach late prophase, we can see distinct chromosomes formed. So basically, it will be two sister chromatids with a centriole in between. So now these are formed, right? And one more thing, if you see the nuclear envelope here, right? It has started to disintegrate, right? Here. So if we come to the next stage now after that, so what happens in metaphase? Let's see that what happens in metaphase. So now in metaphase, the first thing that you'll notice is that the nuclear envelope is completely disintegrated, right? So here what first thing is, now nuclear envelope completely disintegrates. What's happen what happens after that, right? So as the nuclear envelope completely disintegrates, the chromosomes which are formed are spread throughout the cytoplasm of the cell, right? So basically, what is happening is that the chromosomes are now completely condensed now, right? And since the nuclear envelope disintegrates, these chromosomes are spread throughout the cytoplasm of the cell. Now these chromosomes are made up of two sister chromatids, right? And these chromatids are held together by a centromere, right? So there is a small disc-shaped structure which is present at the surface of centromere, and these are known as kinetochores. These are known as kinetochores. Course. Now these are the structures which serve as the site of attachment of spindle fibers. So we have the spindle which starts forming during prophase, right? Now these spindle fibers are attached to the centromeres by kinetochores, and they are moved into the position at the center of the cell. Right. So basically, metaphase is characterized by what? it is characterized by all the chromosomes coming to lie at the equator of one chromatid of each chromosome, and the chromosomes are connected through this kinetochore to the spindle fiber from one pole, and sister chromatid is connected by the kinetochore to spindle fiber from the opposite pole, right? So basically, there is a plane of alignment of the chromosomes at metaphase, and this alignment is known as metaphase plate. This alignment is known as metaphase plate, right? So let's see again the key features of metaphase. Let's just again remember the key features of metaphase, right? So one thing is that the spindle fiber attaches itself to the kinetochore of chromosomes, right? So basically the nuclear envelope completely disintegrates, and first the chromosomes are spread throughout the cytoplasm of the cell. Then what is happening? Then we have spindle fibers. They attach to kinetochore of chromosomes, right? And then what is happening? Third is that these chromosomes are moved to spindle equator, and they get aligned along the metaphase plate through spindle fibers to both poles, right? The north and the south pole. So chromosomes are moved to spindle equator, and they get aligned along the metaphase plate through what? Through spindle fibers. Through spindle fibers and to both the poles, right? To both poles. So if you see the diagram as well, guys, if you see this diagram as well, what you are noticing is that during the start of the metaphase, basically during the transition to metaphase, now the nuclear envelope is gone, and that is why we have these chromosomes lying here and there in the cell, right? Then there was an initiation of formation of spindle, right? Mitotic spindle. So now we have spindle fibers formed, and these spindle fibers are basically present towards both the ends, right? So north as well as south, right? Now these are getting attached with the chromosomes through the kinetochores which are present on the centromere, right? And there is an alignment. So during metaphase now, there is an alignment of all of these chromosomes. Here, if you see that all the chromosomes are aligned at basically in one plate. This plate is known as the metaphase plate, and they are basically aligned at the equator, right, to spindle equator. And we basically, if you can see that spindle fibers are joined with the kinetochore at both the ends, and they are present on both the poles. So this basically marks the completion of metaphase stage, right? Now let's see what comes in anaphase. Now, guys, let's see what happens in anaphase. Let's see what is happening in anaphase. So now, once all the chromosomes are aligned in one plate, aligned on one plate on the equator, what happens after that? Let's see that, right? So basically, now as the anaphase starts, right? Now, as the anaphase starts, each chromosome so each chromosomes which are arranged at the metaphase plate, they split. They split simultaneously, right? and the two daughter chromatids which are present are now referred to as basically chromosomes of future daughter nuclei. So what happens is they start migrating towards the opposite poles. So now what we saw in metaphase is that the chromosomes are arranged in on one plate. Now, in the next step, what happens is that these chromosomes start splitting. So they split into basically the future daughter nuclei. They'll be forming the future daughter nuclei, and they begin migrating towards the poles, right? So one will migrate towards this pole and the other one will start migrating towards the other pole, right? Now, as each chromosome moves away from this plate, from this equatorial plate, the centromere of each chromosome is towards the pole, right? So basically, they lead towards the edge, and the arms of the chromosomes, these arms, this is the centromere which is attached. So this will start moving towards the pole, and the arms, this will be somewhat folded behind, and it will be trailing behind the centromere, right? So basically, the anaphase stage is characterized by these events. So first event is the centromeres split, and these chromatids, they separate. The centromeres split and the chromatids separate. And the second stage is where chromatids move towards the opposite poles. They start moving towards the opposite poles, and it will be in such a thing, say these are the spindle fibers. Here will be the centromere, and the arms will be trailing behind. These arms will be trailing behind, something like that, right? Same here, right? So even if you see the diagram here, you'll see that basically here what is happening is now they are moving towards the pole. They are moving towards the pole, and they are attached to the spindle fiber through the kinetochores which is present on the centromere. So it is basically pulling it towards the pole from the centromere. Now the centromere is basically moving towards the poles, and these arms, both of these arms, you can see here as well, they are trailing behind. So these are the characteristic steps of anaphase. Now let's see what happens in the last phase of mitosis. Let's see what happens in telophase. So this is, guys, the final stage of mitosis where the chromosomes that have reached, right, basically which have reached to their respective poles, they decondense and they lose their individuality. So basically, all the chromosomes which have reached toward two opposite poles, now they were condensed chromosomes, right? They were condensed sister chromatids. They start to decondense and they become one. So one side of all the chromosomes become one, and the other side which are present towards the other pole, they decondense and they become one. And that is how we get two different nuclei, right? So basically you cannot see the individual chromosomes, they are no longer visible, and the chromatin material that tends to collect in a mass in the two poles. So one gets collected towards the north pole and the other gets collected towards the south pole. So basically, what are the key features of telophase then? First is that the chromosomes cluster at opposite spindle poles, right? These chromosomes cluster at opposite spindle poles, and now their identity as discrete element, right, that is lost, and their identity as discrete elements is lost. So this is the first. The second one is that now the nuclear envelope that starts assembling around the chromosome clusters. So now basically the nuclear envelope again starts forming. So the nuclear envelope assembles around the chromosome clusters, right? Now the third one is nucleolus Golgi complex and endoplasmic reticulum. They all start to reform. They all start to reform, right? So if you see the diagram as well here, what has happened now that all the chromatids which moved towards the poles, they disintegrated, right? They decondensed. And after decondensing, they basically joined together to form a what? They joined together to form a chromosome cluster. And so this cluster is formed and those individual elements are gone. Then we have, if you see here, this yellow line, right? So basically now the nuclear envelope assembles around the chromosome clusters. So nuclear envelope is formed, right? And after that, what happens is that the other cell organelles which are present in the cytoplasm, like Golgi complex, endoplasmic reticulum, nucleolus, everything starts to reform, right? So this basically is telophase. Now what happens after that? So basically you see that still the cells are not separate, guys. Still we do not have two different cells. So after this, there is a phase which is cytokinesis. We heard of this word in the previous video as well. So cytokinesis is division of cell, right? So cytokinesis happens and because of that the cell divides. So basically mitosis accomplishes not just with segregation of chromosome, right, which is karyokinesis. After that, the cell itself gets divided into two daughter cells by the process which is known as cytokinesis, right? So, with that, the mitosis is complete, right? So, cytokinesis basically happens at the end, of which the cell division is complete. So in an animal cell, what happens is this is basically achieved by the appearance of a furrow. So you can see here in during telophase, there is a furrow formed, right? This is a furrow, right? So the cytokinesis is marked by the cell division is marked by the presence of that furrow in the plasma membrane, and this furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Right? Now, what happens in plant cell? Because they have cell wall as well. So basically, in plant cell, the wall formation starts in the center of the cell. So it's ulta, right? It's just the opposite. This wall formation starts in the center of the cell and it grows outward to meet the existing lateral walls, right? So basically formation of new cell wall begins with formation of a simple precursor which is known as the cell plate. Basically this is the middle lamella between the walls and two adjacent cells. Right? Now, during cytoplasmic division, the organelles like mitochondria, plastids, endoplasmic reticulum, Golgi bodies, all of these organelles get distributed between two daughter cells, right? So basically, guys, there's a condition where in some cases the karyokinesis, the division of daughter nuclei, is not followed by cytokinesis, and that is why we have multinucleate condition arising, which leads to formation of syntium. So this happens in coconut, and that is why we have liquid endosperm in coconut. So if you see the diagram as well, guys, what you see is that here what has happened that there are basically two cells now, one here and one here. Both basically the two cells have divided. They have a nucleus, right? And there is a spindle which can be seen in a newly formed cell. And then they have various organelles as well. They have various organelles as well. And then later this spindle disappears, right? So let's see what is the significance of mitosis. Let's see the significance of mitosis. So the first significance, guys, is that mitosis restores the ratio between the nucleus and the cytoplasm, right? So the nucleocytoplasmic ratio, right, that is restored in case of mitosis. Yeah, right. Now basically mitosis is an equational division, right? There is equal division of the chromosomes, right? So this is restricted to diploid cells only. Remember that this is restricted to diploid cells only. However, in some cases, in lower plants and in some insects, haploid cells also divide by mitosis, right? Now it results in production of diploid daughter cells, right? So they have identical genetic complement. Remember that they have identical genetic complement, right? So the growth of multicellular organisms is basically due to mitosis. The cell growth that results in disturbing the ratio between the nucleus and cytoplasm, and that is why to restore the nucleocytoplasmic ratio, we have cell division through mitosis. The other thing is cell repair, right? So basically, the cells of upper layer of epidermis, the cells of lining of gut, the blood cells, all these are constantly replaced, right? Now, basically, it happens through mitosis. So all the functions of cell repairing happens through mitosis. Now also the mitotic divisions happens in the meristematic tissues. So if you see the apical or lateral cambium, the meristematic tissues, all these result in a continuous growth of plants throughout their life, and they follow the principle of mitosis. They divide through mitosis. So guys, there are major significance of mitosis basically, and it plays a very important role in all the important processes of the body, right? So with this, we have finished mitosis. We have covered all the stages of mitosis. I hope you would have understood this video very well. If not, please drop your queries in the comments. Please do mention your queries, your questions in the comments, and I'll answer that. You can also message me with all your questions on WhatsApp. The WhatsApp number is given in the description of the video. And guys, yes, if you like the video, please do like it and share it with your friends. And yes, if you're new to the channel, please do subscribe to the channel and press the bell icon to get all the updates. Okay, guys, see you in the next video. Thanks for watching. Bye bye.